Okay, another function of proteins, again, is the regulation. For example, in hormone regulation, no, molecules, its target cell changes its cellular function. So you can find that in, for example, in regulating the blood glucose level, we have the insulin and glucagon. No, those are peptide hormones, a smaller variety of a protein. Then we have the growth hormone. It simulates cell growth and division. There is another function of protein, which is the transport. So many proteins functions as carriers of molecules or ions across the membranes or between the cells. So you can find this in cell membranes. No? For example, you have the sodium, potassium ATPase, and glucose transport. You can find that in the metabolism, in the biosynthesis of the biomolecules. You can also find hemoglobin for transport. It carries oxygen with it from the lungs to the different organs. Now, you also have lipoproteins, LDL and HDL, high density lipoproteins, and low density hyperproteins, which transport lipids from the liver and intestines to other organs. So the classifications of protein, they can be classified according to its shape or according to its composition. We can have fibrous proteins, the one that would have a long rod-shaped molecules that are insoluble in water. So for example, of proteins that are fibrous, mostly they are insoluble, or keratins, no? You can find your hair, it's not soluble in water. No, they are insoluble. So those are fibrous proteins, and why it will be insoluble can be found in its structure. The second classification of proteins according to shape, if you have fibrous proteins, there is also what we call as globular proteins. So the globular proteins are from the word globular. They are compact spherical molecules that are usually water soluble. And they always have dynamic functions. Daghan kay sila og functions. So for example, enzymes, nearly all enzymes are globular. No? Everyday enzymes are always involved in the biological reactions. We also have immunoglobulins, gamay-gamay lang introduction of foreign invaders in our systems. Our system already produce immunoglobulins, and of course we have hemoglobin, the oxygen carrier to the blood. Okay, the hemoglobin are also globular proteins. Also the albumin, they are carrier of fatty acids in the blood. They are also globular. Simple proteins. So the proteins can be classified according to two compositions. So we can find simple proteins and conjugated proteins. Simple proteins contains only amino acids, and conjugated proteins consist of a protein and the non-protein component, or we call the non-protein component the prosthetic group. For example, there is a glycoprotein. So protein siya, but there is a carbohydrate component. Manay tawagon siya glycoprotein. Lipoprotein is protein with lipid molecules also in the structure. Metalloproteins, when it's a protein, but there's also a metal part in the components. The example is the hemoglobin. There is an iron in the part of hemoglobin. We also have phosphoprotein, protein with a phosphate group, and hemoprotein, and this is the one that I have mentioned particular in metalloproteins. It contains the hemi group. We can find this again, as I mentioned, in hemoglobin and also with chlorophyll structure. So let's look at the smaller units of proteins, talk about the amino acids and the peptides. So amino acids talk about the structure, stereochemistry, and acid-base properties, the peptide structure, biological activities, and prelude to proteins hierarchy of protein structure. So here I'm showing you the amino acid structure. In the amino acid structure, there is a chiral center carbon bonded to COOH and NH2. Okay. And then there is a hydrogen and there is an R. Now, usually this is a COOH and this is an NH2, but in water in solution, the COOH will always dissociate to COO minus and the NH2 will dissociate to form or react with water to form NH3 plus. So again, when you look at the whole structure of amino acid, it is neutral, positive and negative. Nitrogen will have positive charge, COO, H, C double bond O, C double bond O. This is the carboxylic acid, no, C double bond OOH, if you remember. And of course, you should remember, again, when the COOH or the carboxylic acid dissociates in water, it's a weak acid, just like your acetic acid. Now, when it dissociates in water, it will form C double bond O minus. The hydrogen is dissociated and the H plus will react with water to form H3O plus. So when NH2 dissociates in water, NH2 katong amine groups, it will form NH3 plus. Okay. So there's 1, 2, 3, and the R group, side chain. We call this R group with a side chain. So the only repeating unit you can see with an amino acid structure is these three groups, COO, NH3 plus, COO minus, NH3 plus, and the hydrogen. And then we have the R side chain. So the R side chain can be altered, okay? And this is how we classify or how we differentiate the amino acids. So this structure is what we call as the alpha amino acid due to the location of the amino group on the alpha carbon, alpha to the carbonyl. So you have your C double bond OO, this is your carbon. And next to your carbonyl carbon is your carbon that would have the NH3 here. So this is an alpha carbon. In the alpha carbon, there is a connected NH2. The protonated form of NH2 is simply positive here. So this is an alpha amino acid. So if you remember this one, if you remember in your organic chemistry, a carbon center that is bonded to 4 different atoms is a chiral center. And having a chiral center, we can expect our amino acid to be to have a mirror image when its mirror image will not be superimposable. So for example, let's have our R side here with CH3. When the R is CH3, we call this one as alanine. Okay, so here you can look at the alanine structure here and look at the mirror image. You have this 3D and you have this two-dimensional way of writing the structure, and you have the wedge towards the dark one. It means it's towards you, and the broken line, it means it's away from you. So you look at the mirror image of this alanine, you actually have the structure. And if alanine here is levorotatory, which means it rotates the plane polarized light to the left, its mirror image would then be dextrorotatory, or D. It would rotate the plane polarized light to the left. If you remember your organic chemistry and the stereochemistry part, there is a configuration for this, the RNS system. But here we don't mind the RNS. But if one is R, the other should be S, in the stereochemistry of chiral structures. Okay, so for the standard DNA encoded amino acids found in proteins, it possesses the L stereochemistry. The one in our system is always an L amino acid, the L or levorotatory amino acid. I don't know, just our system, the nature just chooses the L stereochemistry. But the amino acids are occasionally produced by certain organisms, but are not found in their proteins. In our system, when we process DNA, when the proteins process DNA, all the amino acids found in protein possess only the L stereochemistry. It's amazing how in our system, we chooses only one stereochemistry. Imagine this, they all have the same connectivity to carbon, but the only difference is the orientation in space. And biological receptors are actually very, very, how do you say, very particular to the orientation in space. If you remember, if there is only a change in certain orientations of the groups, no, then some biological systems could not recognize them. So also when the proteins that is used in to synthesis, no, the amino acids only utilizes the proteins having amino acids only utilizes the L amino acids, the L with the L stereochemistry. So where is the origin of the L and the D stereochemistry? The L stereochemistry is derived from the L-glyceraldehyde. No, this is our L-glyceraldehyde. You have C2. From now on, since you are in the biochemistry node, always have to remember the short way of writing your functional groups. The CHO is C double bond O hydrogen. CHO, COH is C double bond O. So you always have to remember that carbon has 4 bonds. It doesn't have any 3 bonds. So how could it be attached to 4 bonds? It should be C double bond O and hydrogen. So that will make carbon having 4 bonds. So CHO, that's an aldehyde, and carbon here, that's an alcohol, CH2OH. No, 1, 2, 3 carbons, and there is an OH in the second and third carbon. So this is an aldehyde. The common name is glyceraldehyde. Okay, there is an IUPAC name for this, of course, that ends with aldehyde. 1, 2, 3 propanaldehyde, but you have 2, 3 hydroxy, no, propaldehyde. The common name is L-glyceraldehyde here. So this is the structure of the L-glyceraldehyde. So the OH, when this orientation, the O is to the left, no, and the H to the right when CHO and CH2 is away from you. And this is the opposite one here. So when we are writing on the amino acids, you also have the structure here. You have the carbonyl carbon, no, oriented away from you, and another, the R group, oriented away from you, the broken line hydrogen oriented towards you, the right, and NH3 oriented towards you here. And so similar to the structure of this, or the orientation, the L-glyceraldehyde, that's also how the amino acids was also agreed to be considered an L-oriented, okay? Also for the D amino acid. So we just shown a simple amino acid. We call this one alanine when R is CH3, but of course, there are 20 amino acids. Now we just have to change on this R, okay, and we will actually have other kinds of amino acids. So the 20 standard amino acids can be clustered into groups based on their side chain structure. So the R, kaning R nga atong dimension here, pwede siya maklasify atong amino acids, no? Nga if R is nonpolar aliphatic R groups, or if R contains an aromatic R group. So when you say aliphatic R groups, kung ano siya, mga length chain, CH2, mga inana, no, or CH3. It's like this one. When you say aromatic R groups, na siya benzene ring in it sa R group. Okay? So example, or if the R groups has polar, uncharged, it can also be considered bipolar and uncharged R group, or passively charged or negatively charged. So let's look at the structure here. So you can have, if we have nonpolar aliphatic R groups, so you can only find hydrogen, CH3, CH2, CH2, CH2 here, CH2, CH3, CH2, CH3. Example, you have the glycine, alanine, proline, valine, methionine, isoleucine. So these are the nonpolar aliphatic R groups. These are the amino acids, alpha amino acids. Now you look at the structure here. You always have COO and minus and NH3 plus and hydrogen, COO minus NH3 plus. Okay, you always have this 3 structure. When you look at here, this you also have three structure, only that there is a cyclic ring formation with the R group. So you have the basic structure, three groups here. The only one that's being changed or altered are the R groups. Okay, you have the carbon-carbon chain here in the R groups. You also have, when you say polar, when usually it contains in the R group, there is an oxygen or there is a sulfur or amine, C amide attached to it. So for example, serine, threonine, cysteine, asparagine, glutamine. Okay, you don't need to memorize all this, but you just have to familiarize with the structure. Then you can also have an R group with an aromatic ring. You have here CH2, but there is a ring, this phenylalanine, and there's an OH, it's tyrosine. And there is a ring here, CH2 and H double bond here, it's tryptophan. Now, for tryptophan, for this aromatic R groups, it's easy actually for them to be analyzed, no, in DUV, because the aromatic groups absorbs light. That's an analytical method of detecting our amino acids and proteins. The presence of the aromatic amino acids can be easily analyzed with UV. Then we can also have an R group that is passively charged. Asa man nato na makita ng passively charged? Whenever you have an amine in the ring, no? So na siya NH2, again, when NH2 dissociates in water, okay, it will always form NH3. No, if you remember, kasi nakalimot naman sa inyong chemistry, no? When you have makita ninyo?
Yes, ma'am.
I hope naka-record ko. Naka-record ba ko?
Yes, ma'am.
Okay, let's have it here. For example, you have, remember your CH3. Okay, for example, you have your CH3, COOH, no, C double bond. Excuse me. Okay, so let's write the structure. Okay, let's write this. It's funny. Anyway, you have your CH3, CH3, C, O. When you have COH, COOH plus water. Okay, just an example, when you have COH, it will dissociate to form, like this is your R and you have your COO minus plus water. Plus water plus H plus. And H plus will react with water to form HCO plus. Whenever you have like NH3, like your ammonia and water, okay, it will dissociate to form NH4 plus, and of course the OH, no? The OH. So that's why when you meet structures like R, say RNH2, like when you have RNH2 also, NH2 in water, okay, it will react to form RNH3 plus and OH. Okay, so let's wait for it. Okay, so back to the discussion. I hope that you don't mind the background now. Okay, so you can also have an R group. That is for your amino. When you have an amine group, for your amine group, it will always be passively charged, in water. The amine groups will always be reacting with water to form NH3 plus. So all the amine groups will be passively charged. Exactly. Here you have NH2. So when nitrogen would have 4 bonds, when nitrogen would have 4 bonds, no, it will always be passively charged. So count on the number of bonds for nitrogen, and then nitrogen will be passively charged. So you can also have negatively charged group when you have carboxylic functional groups in the structure of the side chain. For example, cetyl bond OO, it doesn't in water, it wouldn't assume the structure of excuse me, COOH. It will always assume that C double bond OO minus. So again, when you have COOH, carbon would have 4 bonds, 2 double bond with oxygen and a single bond with another oxygen, no hydrogen. So the COH will not be COH in water, but it will always be dissociated to form COO minus as H plus. So this will be the assumed structure for your amino acids having negatively charged R groups. For cysteine, the one that contains the sulfur, SH, no, CH2SH, this is your cysteine. It can also undergo oxidation and reduction to form disulfide bonds. So this is a diamino acid cysteine. So when 2 moles of cysteine, SCYSTAIN, this is your amino acid, it can be oxidized to form cysteine, CST, without E here, no, with a disulfide bond. You can find this kind of reaction kanang magpakulot mo, no? Magpakulot mo, then it will form disulfide bond. You have this structure on your hair, no? For proteins, the keratins in your hair will be oxidized to form this kind of bond. So, makulot na dayon. It's a chemical reaction going on. There are also non-standard and modified amino acids. The one that's being modified amino acid, but you can have this, for example, you can look at the structure whenever you have carbon and you have your NH here, and then your hydrogen, of course, and your COH. There are the COH here. NHCH. So this is your COH, hydrogen, and NH here. So this is an example of hydroxytoluene. So carbon, hydrogen, CO, NH3, and then this is R group. It's not in the 20 amino acids, no? So that's why it's called non-standard and modified amino acids. Sometimes it can be found, but not all the time. The usual 20 amino acids that's found in proteins and in the synthesis, biosynthesis of nucleic acids are the 20 amino acids that I have mentioned earlier. Okay, but just know that there are non-standard and modified amino acids that can also exist. So amino acids are charged or ionized under cellular conditions. It's the one that I've shown you earlier. So the non-ionic form of amino acid is this: C double bond O, OH, and you have your alpha carbon here, your carbon here, next to your COO, carbonyl carbon. And then you have your NH2 and H and R. So one group, COH and NH2, and then R. So you notice that there is no charge on your carboxylic acid and your amino group. This is what you call as the non-ionic form. But again, as I've mentioned in the reaction earlier, that all carboxylic acids and amines, when in water, they will dissociate. The carboxylic acid will dissociate to form COO minus, C double bond OO minus, and the NH2 will react in water to form NH plus. And this is why amino acids are charged under cellular conditions. So when you have COH and NH3, you call this one as white ionic form. Your amino acid is still neutral, but they are charged. They are ions in under cellular conditions. That's why when I presented the amino acids, I presented in the white ionic form, because in reality, the amino acids exist in white ionic form. So when you have, again, COH and H3 plus, those are white ionic forms. So your amino acids pH can be altered, by changing on the acidity of the solution. So for example, you have here your amino acid with your glycine here, your R here for glycine, the simplest amino acid. You have NH3 plus COH, NH2. Kaya dito mag-start na lang. NH3 plus H2 and COO. The R group is H. So the simple amino acid is the glycine. So when we alter the pH, okay, you can actually alter on the charge of your amino acid. So for example, start with NH3 plus here. When we dissociate COH, when we deprotonate our amino acids, you can have COO minus, and then you have your NH3 plus. And if further deprotonate, then you will have your NH2 CH2COOH. This white ionic form is when you have a neutral charged amino acids. But then there is a point when the pH at which the amino acids is and intermediate. The isoelectronic point is the pH at which the amino acid has a net charge of 0. And that point, the pH at which the amino acid has a net charge of 0, this is the zwitterionic form. Meaning, for example, here in amino acid, okay, so sorry for the interruption. I've been moving around the house now because there's a construction going on. And so I'm now in the car now to avoid the sounds. So anyway, going back, where am I? Okay, so again, when we have these ionic forms, it is when the pH at which the amino acid has a net charge of 0. The ionic forms is also the isoelectronic point of the amino acid. So the isoelectric point, rather, or the PI, is the pH at which the amino acid has a net charge of 0. No, so when we alter, for example, for glycine, the PI is 5.97, which also means that the pH at which this amino acid glycine will be, the pH at which this white-ternate glycine to be 0, or the net charge to be 0, the pH will be 5.97. When we increase the pH of the solution, okay, so when we add more hydroxide to the water, to the solution, what will happen is the pKa will be shifted and the forms of the amino acids will also be altered. So when we make our OH, when we increase on the OH concentration, for example, what happens is the NH3 here will be deprotonated rather, and the structure of the amino acid will be changed also. So what happens is there will be a net charge that is negatively charged when we increase the pH of the solution. When we increase the pH of the solution, the hydroxide ion concentration will also increase. Remember your general chemistry. Now, from the isoelectric point, Pi, in the zwitterionic form, when we decrease the pH, which means that there is a greater hydronium ion concentration. So we have more H plus or hydronium ion concentration. So what happens is it can react with H plus to form COOH. So at a lower pH or at a lower hydroxide ion concentration, the form of the amino acid will also be altered and the net charge will be positive. So at a lower pH and lower hydroxide concentration, the net charge is positive. At a higher hydroxide concentration at a higher pH, the net charge is negative. But only at the Pi, or the isoelectric point, the pH at which the amino acid has a net charge of 0. Okay, so that's why the amino acids and the proteins are pH dependent. So when amino acids reacts, with another amino acids, they call a bond, they would form a bond called peptides. So peptides is when the amino acids, what is a peptide? When the carboxylic acids and the amine groups of another amino acids would form a bond to form an amide. C double bond O NH2. This is an amide structure. And this is what we call as a peptide bond. So the C, the OH, and the hydrogen of the amine groups of the second amino acid will be removed. No? So the forward reaction, water is removed and you form a peptide. So this kind of process, when the when the first amino acid, the carboxylic group of the first amino acid and the amine group of the second amino acid will react, it will form a peptide bond. The peptide bond again is an amide bond, C double bond O N H. This is an amide. And since water is removed, this is a reaction that is called a dehydration reaction. Water is removed. In the same way, when you have a peptide, okay, two amino acids is being connected by a peptide bond, we can also reverse the reaction back to its amino acid. So how do we do that? By including water. And that's what we call as when we integrate water into the reaction to form your two different amino acids, it's a hydrolysis kind of reaction. No? We return back the OH or water into our individual amino acids. Okay, so this is how the linkage of amino acids would form peptides. So it's also possible not just that two amino acids would react to form peptide. So if there's more than two amino acids being involved, then you have a polypeptide structure. Okay, here you have C double bond O and H. So these are your peptide bonds, or in particular, the peptide bonds is just made up of amide bonds. No? So how many amino acids in the structures? You can have 1, 2, 3, 4, and 5 amino acid structures. So when there are more than 2 amino acids in the structure, you call this one as polypeptide. This one is what we call as a dipeptide. So when we write on the polypeptide structures, no, we start from the left side with the N terminus or the amino terminal end, and the right side to be carboxyl terminal end structure here. So N to the C, N terminus or to the C structure. So there are 3 types of bonds found along the backbone structure, peptide backbone structure, repeated over and over. You have your carbon from the C double bond O, and then you have your carbon nitrogen from your amide and your nitrogen and from your alpha carbon here. Okay. And since you see this one to be C double bond O, nitrogen, of course, if you remember your organic chemistry, whenever carbon has a double bond, no, and bonded to 3 types of atoms, 3 atoms, your carbon will assume a what shape? Anyone? Remember the geometry? Unsa nga shape ang i-assume ni carbon? Anyone? Ay, naminos ako. So your carbon will assume the trigonal planar structure, C double bond O, carbon nitrogen structure here. Okay. So the planarity and rigidity of the amide or the peptide bond is due to the resonance structure. No? So remember you have your carbon double bond O carbon. So this is your amide.